بسم الله الرحمن الرحيم. السلام عليكم ورحمة الله وبركاته. إن شاء الله دي أول محاضرة عن جزء السيدز أو البذور. السلام عليكم ورحمة الله وبركاته. إن شاء الله هنتكلم على محاضرات في السيدز أو البذور. Seed defined as mature fertilized ovule adapted for propagation and distribution of plant. So seeds are originate from ovules after fertilization of the ovules and ripening of the zygote or fertilized ovule into fruit containing seed inside it. Seed containing embryo, which is responsible for production of new plant. So seeds are function in distribution and propagation or multiplication and reproduction of plants. Growth or origin of seeds in the ovary, they exist as a small projection or attachment on placenta inside the ovary. There are typical layers or typical elements forming the seed. Here diagram illustrating fruit wall and the seed inside it. This is a fruit wall and the seed inside it. There are two cotyledons of dicot plant. Plant is coffee bean. So outer coat or outer layer of the seed is known as testa or outer seed coat, which is one or two coats originating from the integument of ovule. We will come to define integument. After testa we have perisperm, which formed from the tissue of the nucellus. Endosperm formed from the tissues of the nucellus surrounding the embryo, containing reserve food material as aileron grain or fixed oil globules. And finally, the innermost layer is the ovary, which originates from zygote after fertilization of ovule. So this is endosperm. This is outer seed coat, perisperm, and endosperm, and the embryo. This is the fruit wall, epicarp, mesocarp, and endocarp or inner fruit wall. From the morphological point of view, seed, we will notice that the seed has shape. The shape may be spherical, rounded, kidneyform, or renal, or reniform, maybe flattened, maybe disc shape. Different shapes of the seed exist in different plants. The outer seed coat or the surface of the seed may be smooth, may be pitted, may have reticulated structure, and may be hairy. Testa may be membranous, as a thick membrane, or thin, may be woody with lignified tissue, sometimes could be fleshy. There are different marks or scars on the outer seed coat or the testa. The first is the hilum, which is a scar left at the point of attachment of the seed to the funicle or the carrier of the ovule, or the maybe opposite to the stalk of the ovule. So attachment of the funicle to the ovule and introduction of vessels for nutrition or vascular bundle for nutrition. There is a mark marking the hilum. Microbile, second scar or the second mark, which is a small hole on the top or the upper part of the ovule, which marking the passage of the pollen tube and containing the pollen grains to fertilize the ovule. The raphe, which is narrow ridge running from the hilum to the point marking attachment between the ovule and funicle. So attachment of the funicle and the ovule wall will change the type of structure of the tissues in this attached area, forming a raphe. This is the funicle, the ovule, micropyle, introduction of the pollen tube containing the pollen grains to fertilize the ovule. Raphe, if the ovule is fused with the funicle. And the hilum, marking attachment or entrance of vascular bundle from the hilum to inside the ovule. As a mature ovule, there is typical structure or typical layers. Ovule is made up of nucellus and integuments or ovule walls or ovule coats. The nucellus is largest part of the ovule, containing the embryo sac as well as nutritive tissue, and remain present in some flowering plants after fertilization as a source or as a reservoir for nutrients to provide embryo with nutrients. This is nucellus, which is the main part of the embryo, containing nutritive or stored nutrition for the embryo. The integuments or ovule coat or protective layer. Usually we have one integument in gymnosperm, that is a diagram, one coat, one protective layer surrounding nucellus, which is afterwards forming the embryo. And in angiosperm we have two coats or two protective layers. This is one and another integument. The integuments enclosing nucellus except for a small gap marking the microbile, which, as we said, it's the entrance of the pollen tube containing the pollen grain to fertilize the ovule inside. Another part of the ovule, which is called the claza, it is the basal swollen part of the nucellus, and from which start integuments or protective coats surrounding the embryo to grow. As is the diagram, this is the claza and nucellus, embryo, and embryo coats or protective coats or the integuments. And here fusion of the ovule with the funicle forming raphe, opening of the micropyle. According to direction of growth of the ovule or manner of growth of the ovule, you have different types of ovule. First type is the atropous or orthotropous ovule, which is erect or straight type of ovules, in which having the funicle, claza, and micropyle at one and straight line, showing no raphe. It is straight or erect. This is funicle, here the hilum, micropyle, and lower part of the nucellus, which is claza. Micropyle, claza, and hilum, all three structure lying on the straight line, so it is straight or erect type of ovule. The opposite is anatropous ovule, in which the ovule is inverted or bent on the funicle and fuse with it. So this is ovule and funicle, so it is converted and bent and fused with the funicle. So this is a new structure arise due to fusion between the ovule and the funicle, which is the raphe. The micropyle will be near to hilum, and the formed raphe running from one end to the another end of the ovule. Amphitropous, in which the ovule is partly bent on the funicle, so micropyle, claza are at one line at right angle to the hilum. It is partially bent with the funicle, so that there is a small raphe, micropyle, claza, and here is the hilum. A straight line perpendicular or at right angle with the hilum. Here, as we said, the raphe running from the claza to half distance of the micropyle. Short distance, the claza, and here the micropyle. Short distance between the claza and micropyle, maybe half the distance between them. In all the previous three types, which is atropous or anatropous or amphiotropous, the nucellus is straight, characterized by being straight. Growth normally without bending or overgrowth of the nucellus. Fourth type is the campylotropous ovule, in which the ovule bent on itself. Here in the amphitropous, the ovule bent on the funicle. But the campylotropous, the ovule bent on itself, or there is overgrowth in one side of the ovule and other side the growth is normal. So this is enlarged growth of tissue on one side, or the ovule will, as we said, bent on itself. So the micropyle, clasa, and hilum will be close to one another as the tissues pushing each other. So the micropyle will be near the hilum, and the clasa, both are near to each other. As we said, we have different shapes and form of the seed, with the spherical, ring shape, disc shape, or flattened. Size of the seeds may be very small as in mustard, or very large as a Ludwika seed, which may reach up to 15 kilograms. Histologically, the seed structure, we have outer seed coat and different layers of the outer seed coat or the testa. First layer is epidermis, which is definite layer, sometimes with characteristic features. This layer may be normal polygonal tabular parenchyma cell, or maybe palisade-like cells as in fenugreek. Sometimes may be formed of lignified sclerites as in Nux vomica, and sometimes bearing hair as in Strophanthus. Below the epidermis there is hypodermis or subepidermal layer, which may be formed of basket-like cells as in fenugreek, or large or giant cells as in black mustard, large collenchyma cells as in linseed. The inner seed coat or the inner part of the testa may be formed of sclerenchyma layer, or hyaline layer or nutritive layer, which contain reserve food material and exhausted, so leaving collapsed cells, and containing the pigment layer, giving the seed its characteristic color. In addition, sometimes some seeds have a perisperm or structure called perisperm, which is membranous coat surrounding the embryo. The endosperm surrounding also the embryo and containing a long grain or fixed oil globules for nutrition of the embryo. Sometimes containing starch, and it is described as starchy or farinaceous endosperm, or containing oil, fixed oil, and this described as fleshy endosperm, or containing mucilage and described as mucilaginous endosperm. The embryo consisting of one in monocot plants or two in dicot plants, one cotyledon or two cotyledons, and the radicle. The cotyledons or the embryo leaves containing the reserve food material and forming the first leaf after growth of the seed. The radicle give rise to the first root arise from the seed after growth. We have different shapes of the embryo according to arrangement or direction of the cotyledon and the radicle. This diagrammatic illustration of the seed coat layers. The epidermis followed by hypodermis and some seed covered with cuticle, large tabular cell, or followed by parenchyma or collenchyma cell, sometimes sclerenchyma cells, lignified, nutritive or hyaline layer formed of collapsed cells after exhaustion of the reserve food material contained before. And last layer is the pigment layer, followed by the endosperm, either long grain and fixed oil, and followed by the elongated or columnar cells of the embryo containing also a long grain and fixed oil globules. Inside the testa or the seed coat we have kernel structure, which is formed of the endosperm and embryo. So if we have endosperm, this seed is described as albuminous seed, in which embryo is surrounded by the endosperm or endosperm and perisperm. Either both of the endosperm and embryo are formed of thin mold polygonal cells containing their food material of long grain and oil globules, as we said before. Or if the endosperm is absent, so the seed described as X-albuminous seed, which is formed of embryo alone at the inner tissue, and the seed coat or the testa formed of different layers forming the seed coat. Sometimes the seeds or the seed coat develop outgrowth during its development with different characteristic shapes according to the type of the plant. First outgrowth is the arillus or aril. This outgrowth arises from the funicle or the hilum tissue and cover the whole or part of the seed, as in cardamom or nutmeg. This is shape of the aril arising from the funicle or hilum and covering the seed completely or part of the seed. Aril load, which arises from the tissue of the micropyle, cover part of all of the seed. So both structures covering part or all of the seed, but the origin in arillus derived from the funicle or the hilum, while in aril load derived only from the micropyle or the tissues of the micropyle. Sometimes develop from both funicle and micropyle. So either micropyle alone or micropyle and funicle, but in arillus, hilum or hilum and funicle. Third outgrowth is the carnicle, which is fleshy outgrowth formed from the tissues near the micropyle and cover it. So this is around or the tissues surrounding the micropyle and itself or this outgrowth covering the micropyle. So this is a carnicle, as in castor seed. The strifule, which is local enlargement over the brachy, outgrowth from the raphe, as in colchicum. Wing, which is membranous extension of the testa, any part of the testa, as the form of wing, as in strophanthus. Plum, which are group of hairs on the top of the seed. Own, which is group of hairs borne on cylinder stalk, or plum borne on cylinder stalk, as in strophanthus. This is stalk for the group of hairs of tuft of hairs, or the plum as described before. If we have LS in the seed, there is different shapes of embryo according to the development. First type is a straight embryo, in which cotyledon and radicle are on a straight line. This is longitudinal section parallel to the cotyledon and longitudinal section perpendicular to the cotyledons. Two cotyledons, one radicle, here the cotyledon and radicle on one line. Curved, in which the radicle start to curve or having slight curve in front of the cotyledon. Coiled, when the cotyledon having spiral or coiled development or growth with against straight embryo, as in datura and hops. Bent, when the radicle or the cotyledons bend on itself, we have incumbent or accumbent type. In the incumbent type, the radicle bent against one of the cotyledons in dicot plants, as in cannabis. Here, radicle, the cotyledon facing the radicle, and other cotyledon away from the radicle. This will be more clear here. One cotyledon, the other, and the radicle. In the incumbent type, the radicle bent against two or facing both the cotyledons in dicot plants. Here the radicle, one cotyledon, and the other. So if the radicle bent in the direction of the cotyledons, if it is only facing or in front of one cotyledon, this is incumbent type, and if facing both the cotyledons, sometimes in between the cotyledons or facing the edge of the two cotyledons, as in fenugreek, so this is accumbent type of embryo. Thank you and good luck.